بسم الله الرحمن الرحيم. السلام عليكم. احب اعرفكم عن نفسي. انا علي باسم، طالب في كلية الصيدلة-جامعة بغداد، مرحلة ثالثة PharmD. اليوم ان شاء الله فيديو مالتنا حيكون ملخص لبحث coffee consumption by type and risk of digestive cancer, large prospective cohort study. طبعا the study was done by the authors where these the following. Research was from the UK by this journal and published by Nature Publisher Group. طبعا presented by me علي باسم وزميلي غياث عبد السلام احمد. كذا وشوي البرزنتيشن اسمها مقلوبه، لكن علي باسم وغياث عبد السلام احمد. Okay, as it's known, coffee is one of the most large consumed beverages worldwide. ولو احنا عندنا بالعاب صراحه الشاي يغلب القهوه، لكن mostly worldwide in coffee. There are more than one type of coffee: instant, ground, and decaffeinated. What are the differences between them? Well, instant coffees are made in factories where they are processed and, let's say, grinded into patches and salted, where you can just put them in hot water and they will, let's say, turn into coffee. Okay, while ground coffee are the same coffee beans where you need to put them in a machine and grind them in order for you to get coffee. Well, you might ask, what's the difference between them? There are a lot of differences in their chemical structure, mainly go to their chemical process of making them and using the preservatives and whatnot. Okay, what is the significance of this study? Why are we doing this study? We are doing this study because many studies previously done this subject, but the problem that they did make it depending on only one type of coffee. They did not take, for example, instant to ground decaffeinated and compare them between each other. This is why the objective of our study is to investigate the association between coffee consumption and the risk of digestive cancer between multiple types of coffee and comparing them with each other. Okay, what did past observation? As we said, there are a lot of, let's say, observation made before by many publishers, many researchers. What did they say? Well, the past observations said that there are inversely association between liver cancer and consumption of coffee. By means of increasing coffee consumption, decrease the risk of liver cancer. But for other GIT cancers, it's not that really clear. There are many conflicting researches, let's say. Okay. Now, we said there are a lot of differences between instant and ground coffee. What are these? For example, one of them are acrylamide, which is considered as type group 2A as a probable human carcinogen. Well, it's been also conflicted. Some say it is, some say it's not. But it's also, let's say, honorable to mention that it's considered as a carcinogen. And it's also, if I forgot to mention, it's twice its concentration in instant coffee than it is as in ground coffee. Okay, methods. How are the data being collected? The data are being collected by a UK biobank, which is a bank that contains many information of patients or other volunteers for their, let's say, life and their health informations. They are recruited from 2006 to 2010, 4-year, 500,000 volunteers between the ages of 40 to 69, which is the design of the study. Prospective study excluded two types of people who already has previous cancer and been treated, or the patients who had cancer in the first year. Remember, we said it lasted for 4 years. So the patients who had cancer for 1 year have been, let's say, eliminated from the research. Also, the exposure, how are the data being collected? The data are being collected by the patients themselves, who given information about the cups per day and also most common type consumed, as we said, decaffeinated, instant and ground coffee. And if I forget to mention, what is decaffeinated coffee? It is a coffee that has been removed approximately 79% of its caffeine by chemical procedure. Okay, last but not least, what are the covariants of our research? Age, sex, male and female, education, socioeconomic, deprivation, body mass index, alcohol consumption, smoking, physical activity, vegetable and tea intake, comorbidities, high cholesterol and diabetes. All of these are covariants that we take in consideration in our research to make sure that there is no bias in the research. What is the analysis? We use more than one type of a statistical analysis, which is one of them is Cox regression, which give you two variables and their relationship, if there's a relationship between them with a certain time, which is age in our study, let's say. Also, dose response analysis, sensitivity analysis, excluded the first 2 years. Remember, we said there are 4 years for the study. They did exclude the first 2 years. So you have 3 exclude. The first 2 years for observation, and also you have excluded the patient who have already cancer and the patient who got cancer in the first year. and also a test for heterogeneity by coffee type. شكرا جزيلا. تحكم زميلك.
السلام عليكم. My name is غياث السلام احمد, and I will continue with our presentation about the correlation between coffee consumption and cancer. As my partner, I just explained to you the data and the introduction and the actual methodology of how we are going to conduct our study. Next up, we are going to actually look at the results that we collected. So first of all, the first type of results that we collected are the baseline characteristics of our sample population. So we asked our participants about their coffee intake, how much they consumed coffee by the number of cups per day. We also collected their age, their sex, their background, their education. And the main results that are worth highlighting are the following. 77.4% reported that actually consuming coffee, whereas about 22% did not consume any coffee from our sample. And also, coffee users in general tended to be older, tended to be male, and also of higher educating background. And last but not least, some factors like alcohol intake we noticed were associated with a greater coffee intake, but however, something like smoking status did not really have much effect on the coffee consumption. So after we've actually got to know our sample population, next we're actually going to look at the main data and the results of our research, those being how coffee consumption correlates to cancer. So we're going to as we look at these results, we notice, and this is the primary result of our study, that coffee intake has a protective effect against liver cancer. What this means, an adjusted hazard ratio of approximately 0.5% for coffee drinkers. This means that coffee intake actually almost halved the cancer risk. So as it decreased it, we also look at results from other types of cancers, mainly for esophageal squamous carcinoma. We noticed from our study that coffee intake increased the risk of this type of cancer, although this type of cancer actually in previous studies had conflicting results or there was no association seen between coffee intake and this type of cancer. But as for other types of cancer, we didn't actually see a significant correlation, and so our main result remains for the hepatocellular carcinoma. And also, as my partner actually explained to you, that there are differences between the different types of coffee, which actually affects their biochemical constituents. This may lead to actually affecting which types of coffee produce which effects on cancer incidence. The main result being that instant coffee had the greatest inverse association with hepatocellular carcinoma, whereas ground and caffeinated coffee also had a protective effect against hepatocellular carcinoma. However, this effect was not as substantial or not as significant. And finally, decaffeinated coffee even suggested an increased risk of some types of cancer, these being gallbladder and extrahepatic bile duct carcinoma, and these are the specific data. These are, as we just said, these are the main results of these are the main findings and conclusions of our study. Protection against hepatocellular carcinoma and increased protection the more we increase our coffee intake, but not much data or correlation for other types of cancer. Now, of course, every study is going to have its limitations, and our study is no exception, be it because of lack of resources or because of lack of accessibility to more kinds of data. So the main types of limitations that our study had are exposure misclassification. So for coffee consumption, we only took the data for coffee consumption at the beginning of the study. And so if the coffee intake by our participants changed over time, this is not going to be reflected in our data. However, this is not a problem, hopefully, because previous research has indicated that adult coffee consumption is generally stable over time. Second of all, small number of certain types of cancer, and like small intestine, intrahepatic bile duct carcinoma. Although we had a large sample in general, but the number of cancer cases was not as high, and so this may actually not make the associations with these cancers, these rare types, as clear. And finally, there are some unmeasured confounders. We couldn't get data on the H. pylori infection for our participants. We didn't get data about the effects that additives to coffee had, like milk, non-dairy creamer, and sweeteners. If these confounders had effects, we could not measure them. And this leads us to our next point, recommendations. We know that there is a correlation between coffee consumption and cancer, but we don't actually know the mechanism, and so we suggest mechanistic studies to look at what exactly is causing these effects, what is causing this protective effect, these potential increased risks. And this could be to potential biological mechanisms like to the antioxidants and chlorogenic acid or caffeine that coffee contains. And next up, as we said, there could be some coffee consumption misclassification, and so if future studies could actually get a detailed data on the coffee consumption, this could actually confer more rigorous property to the research. And finally, extended cohorts. Our sample was confined to the UK Biobank sample. If there are more differences for other populations, this may not be reflected here. And thank you very much for listening. This was our study. I hope you learned something, had some new insights for coffee, which of course almost everybody has consumed. And thank you very much for listening.